Hello and welcome to C1.1 Enzymes and Metabolism. This is probably one of the fundamental topics within IB biology because it crops up in so many of the other topics. So you kind of need to know this before you do many other topics that all include the use of enzymes or enzyme controlled reactions. We've got the biology guide here for SL, and we've also got some HL content if you want to have a read through there or refer back to it. Okay, so let's think about how you can turn information in this video or literally any video you watch on YouTube into a grade 7. So just watching the video wouldn't really class as revision because it's too passive. If you're watching a video and looking at the information, listening to the information, you need to be doing something with that. So you need to take notes from videos or draw diagrams from videos. The videos that you watch, you should be stopping and starting. Stop it, go back, watch it again, make sure that it makes sense in your head. Write some notes down for that, watch the next bit, not just passively watching it whilst you're scrolling on your phone in the background. Make flashcards for key terminology. So I outline the key terminology in all of the videos, and that's for you as a tool for you to use and make sure that you know what those terms mean and that you could use them in an exam question answer. Draw and annotate diagrams. I'm really visual. I like pictures. And once you've drawn and annotated a diagram, do it again, and then do it again, and then do it again until you know it. Often in the extended response questions, you can draw and annotate diagrams and you will get all of the marks if you do so. Make revision posters of complex concepts, stick them up, not just in your bedroom, around your house, take them with you, stick them up in the bathroom when you're in the shower. Stick them up next to the TV so that when you're watching TV you can just glance at it, be savvy with your revision. Use the revision platforms that your school's signed up to. So for example, Cognity is a really good one. There's so many questions on there, exam questions, multiple choice questions, questions at the end of every page. Your school's probably signed up to some sort of platform. Use it because you're getting it for free. Watch different videos. So the way that one person explains something isn't necessarily going to be the best way for you to understand it. So if you don't understand it the first time in one video, go and find another video with somebody who explains it in a slightly different way. And the same for at school. If you haven't understood the way that your teacher's taught something, go and find another biology teacher and ask them to explain it. Go and find another student and ask them to explain it, because the wording that they use might be slightly different and might just click in your head. And also practice exam questions. Now I appreciate that there is minimal exam questions for the new spec at the minute. That will increase as we go through the years, but right now there's not very many. Ask your teacher for past exam questions from previous specs. So things like data analysis questions, that's a skill, that's not knowledge. You can practice those so that you practice in those skills. There's lots of questions available on platforms such as Cognity that do cover the new spec. So find as many as possible, hassle your teachers until they give you them, and practice them. Use the mark schemes. Don't be nice to yourself when you're marking those questions. Make sure that you are marking harshly and that you're not being overly generous. Okay, key terminology, like I just said, make flashcards of it. Print out the key terms so that you can look at them, get someone to test you, get someone to ask you what they mean. Can you confidently use them in an exam question answer? If you can't, then do some work on it. Make checklists of the key terminology so that you can tick them off as you know them. And then you can go back, you know exactly what you need to do next time. So you're not going over the same work and doing the same thing even though you already know it. Be savvy about your revision. Okay, enzymes. So enzymes and metabolism. So we're going to talk quickly about anabolic and catabolic reactions. So we need to know the definitions for these. So anabolic builds larger molecules from smaller ones. It involves condensation reactions because we're putting things together and therefore water's given off, and it requires energy. Catabolic reactions is the breakdown of large molecules into smaller ones, involves hydrolysis, so therefore using water, and these release energy. Okay, the basics of enzymes. So you probably should know this from GCSE. Enzymes are catalysts, which means that they speed up the rate of the reaction. And metabolism is all of the chemical reactions within a cell or within an organism. The reactants are the substances that go into the reaction, whilst the products are the substances made in the reaction. Anabolic reactions build more complex molecules, like we just said. Catabolic reactions break large molecules into smaller ones. Globular proteins. So some proteins are globular and some are linear. Linear proteins are usually the ones that don't move around the body. Globular proteins examples are enzymes, and they do move around the body, or they can move around. The enzymes are polypeptide chains, so they're a protein, and they're folded and twisted into a specific 3D tertiary structure. Now, we already know, or we should know if we've done proteins, that protein function is dependent on that protein shape. So that specific 3D tertiary structure is really, really important. It has to fold into the right structure in order for the active site of the enzyme, which you can see just here, for the active site to be a specific shape and complementary shape to the substrate. So your substrate is going to move into that active site of the enzyme. It makes an enzyme substrate complex that's broken down into the two products. So you can see this yellow substrate has been broken down into the blue and the green product. They're the products of the reaction. They will go off and do their job. Okay, so we have a couple of models of enzyme action. So these are kind of theories as to how enzymes actually work. The first is the lock and key model that suggests that the substrate fits into the active site of the enzyme perfectly. So you can see here that it's perfectly complementary, fits into the active site, just like a key fits into a lock. In the induced fit model, this suggests that when the substrate enters the active site, you can see that they're not perfectly complementary. However, once it goes into the active site kind of molds around the substrate to make it totally complementary. Now, it's the induced fit model that we're fairly certain is what happens with enzymes now, but they're just two theories that scientists came up with when they were trying to work out how enzymes completed reactions. We've also got this formula here, so we can see that we've got enzyme plus substrate produces the enzyme substrate complex. This is then going to either turn into the enzyme and the product, or if the substrate comes out of the active site, then it could go back to just being the enzyme and the substrate. And then finally, we've got activation energy. This comes up in multiple choice questions quite often, or just as a 1-mark question. The activation energy is the energy required to start a reaction, and enzymes lower the activation energy. Key buzz phrase there. So the enzymes lower the activation energy. Okay, there's many factors that affect the rate of enzyme-controlled reactions. We're going to have a look at 3 of them here. So we've got the effect of temperature on the enzyme-controlled reactions. Now, as the temperature increases, the kinetic energy increases, and the enzymes and substrates move around more quickly. We then say that this results in more collisions. So we talk about the substrates and the enzymes colliding, and therefore increases the rate of reaction. So when they collide, that means that the substrate has fitted into the active site of the enzyme, and therefore it can be broken down. We can see it here at the optimum temperature. This is the highest rate of reaction. The optimum temperature is not always 37 degrees. So every enzyme has a different optimum temperature. So for example, Taq polymerase is found in hot springs, and its optimum temperature is 72 degrees. So it's not always 37 degrees. Don't make that mistake. So we can see optimum temperature up here, highest rate of reaction. After that, the rate of reaction falls down pretty rapidly as the temperature increases. This is because as we increase the temperature, the molecules move around more, they vibrate more, and therefore some of the bonds within the tertiary structure of the protein begin to break. And this is going to change the shape of the active site. And if we change the shape of the active site, it's no longer complementary to the substrate. Enzyme substrate complexes can't be formed, and therefore the enzyme isn't going to work. We're going to say that the enzyme is denatured. We also have the effect of pH. So enzymes work within a range of pHs. All enzymes work at different pH. We've also got the optimum pH. You can see here, this is the optimum because it's the highest rate of reaction. So for example, pepsin in the stomach has an optimum pH of pH 2 because it's an acidic environment. The enzymes within the small intestine have an optimum pH of around 7, around neutral, because they work in the small intestine. That's their optimum pH. However, they can work at this range. So this enzyme can work from pH 2 up to pH 13, which is a pretty large range. At very low pH and very high pH outside of that range, the interactions within the polypeptides start to break down, and therefore it changes the shape of the active site. So therefore the active site no longer complementary, enzyme substrate complexes can't be made, it lowers the rate of reaction. So we see a low rate of reaction outside the lowest pH in the range, and a low rate of reaction outside the highest pH in the range. We've also got the effect of substrate concentration. So as the substrate concentration increases, so does the rate of reaction. But because there's a limited number of enzymes, what happens is once the enzymes are all working at their optimum and fastest rate, all of the active sites are filled, it doesn't matter how much more substrate you throw at that enzyme, all of the enzymes within the solution, let's say there's 100 of them, all of them are working at their optimum, at their maximum rate. So therefore they cannot work any faster regardless of how much substrate you add in there. So that's why we have a plateau within that graph. You must be able to describe and explain all three of these graphs. You might need to sketch them as well, actually. Okay, we've got intracellular and extracellular reactions. So within intracellular reactions, enzymes remain within a cell and the reaction occurs within the cell. Examples of this include glycolysis and Krebs cycle within respiration. And then some enzymes are secreted and break things down outside of the cell. So examples of this include digestion reactions in the small intestine. So for example, the digestion of starch within the small intestine by pancreatic amylase. In endotherms, metabolic reactions are not 100% efficient. So mammals, birds and some animals use heat produced in order to maintain their body temperature. So any heat that's produced within those reactions, it's used to maintain their body temperature because they maintain a constant internal environment. In metabolic pathways, we've got a couple of diagrams here. So a linear metabolic pathway, you start with one initial substrate, you go through a series of reactions until you get to the end product. So D here is the end product. That is going to be used for something else within the cell or within the body. A is our initial product and we call B and C the intermediates. There will be a different enzyme at each stage that catalyzes that reaction. We also have cyclic metabolic pathways. So we can see we've still got these four intermediates. So A, there's going to be an enzyme that turns that into B, another enzyme that turns that to C, another enzyme that turns that to D, and then we have another enzyme here that will convert D back to A in order for the cycle to continue. So we can have linear pathways or cyclic pathways. Okay, inhibition. So sometimes enzymes can be inhibited, and this might be deliberate. So there's lots of mechanisms in the body that will inhibit enzymes for various reasons in order to speed things up or slow things down. Lots of drugs can be inhibitors as well. So there's lots of practical applications. In competitive inhibition, you can see that the substrate and the inhibitor have a similar shape to one another. So we have to say that they have a similar shape. They don't have the same shape, but they have a similar shape, and that shape is complementary to the active site of the enzyme. So the inhibitor fits into the active site of the enzyme. It means the substrate cannot fit in, and therefore no enzyme substrate complexes can be made, and it lowers the rate of reaction. We've also got non-competitive inhibition. So up here, the substrate and inhibitor are both competing for the active site. In non-competitive inhibition, we have your enzyme and substrate, but then we have this other inhibitor, and this other inhibitor binds to a site away from the active site. We call that an allosteric site. So it binds to an allosteric site, and you can see that it's changed the shape of the active site, so your substrate cannot fit in. So therefore, no enzyme substrate complexes are made, lowers the rate of reaction. End product inhibition. This is where the final product in a metabolic pathway is the inhibitor, and therefore this final product will inhibit the initial enzyme. So it means that the pathway can kind of self-regulate. So if you make loads of of the final product, that will then inhibit the initial, and it will stop that from working for a while. So therefore it regulates the amount. Once there's too little, then the enzyme will start. Too little of the end product, the enzyme starts working again because there's not very much inhibitor. Oh, you can see that there. And then we have mechanism-based inhibition. So this one's slightly more complicated. We know that penicillin can be broken down by the enzyme penicillinase, which is made by some bacteria, and that makes those bacteria resistant to the penicillin. The penicillin inhibits an enzyme called transpeptidase, which is responsible for producing cell walls in the bacteria because it binds to a specific group within the active site of the transpeptidase. Now, scientists have changed the structural makeup of the penicillin to make it able to attach to the transpeptidase, so it will inhibit the production of the cell walls, which will kill the bacteria, but it also can't be broken down by penicillinase. So that's an advantage because you can still administer penicillin. It's not going to be broken down by anything, but it is going to kill the bacteria.